Hello, welcome to our single-cell multiome analysis series with SciDAP. In this last video in the series, we will generate more plots using single-cell manual cell type assignment pipeline. So you select the pipeline from the drop-off menu. I named this sample step 8, cell type, and you select the previous step that was WNN cluster analysis and aggregated sample for this experiment. Then you could select any dimensionality reduction method you like, either WNN, ATAC, or RNA for your plots. You could also provide a clustering resolution and the criteria to split every cluster by group. It could be dataset or condition if you like to generate this kind of plots. You could also generate plots with gene markers and provide genes of interest that will be in your violin plots and UMaps. You also have to provide cell type file. So this pipeline doesn't identify cell types automatically. It creates the new plots with new cluster names. For example, if you need to combine several clusters together, let's say there are clusters that you couldn't identify as a particular cell type, you would give them the same name, and SciDAP will merge the clusters into one. You format this input file like this. You give cluster names manually. You could save it as TSV or CSV file and attach it here. You could also use selected barcodes for your plots or GMT for calculating gene set enrichment. In advance, you could select the color scheme. I'll just leave it classic, but you could play around with different colors. And this will give you pretty much similar results to what we had in WNN cluster analysis, but will generate a few more plots with already annotated cell types. Okay, it's done running. Let's look at the plots. You could notice that every plot that was generated here has the cell types that we provided in the CSV file. You could see that some clusters merged into one, and every matrix that you've seen previously also contains these new cluster names. This is the UMap. Also, in the tab Split by Group, there are all the same plots that you've seen previously. So why is this useful? For example, you run the WNN clustering with the find the gene markers on, so you could identify genes that would actually make sense to provide them as gene of interest in the following step. So here, because this pipeline runs way faster, it is more convenient that you could provide this list of those gene markers which will help you annotate every cluster. And it's the same for the coverage. So these are all ready to publish figures for your paper, and you could download every PNG plot if you'd like. So this was our final video, and you could see now here in the UMap all the cell types together. I hope this tutorial series was helpful for you, and now you are an expert in using SciDAP pipelines. Good luck with your single-cell data analysis.